The thylacine, or Tasmanian tiger, is one of the most exotic and mysterious creatures to have roamed the planet.
It's an incredibly amazing, beautiful marsupial that was brutally hunted by humans to extinction.
But now a world first.
This will be the foundation of bringing the thylacine back.
A team of Australian scientists, in collaboration with a US genetic engineering company, are promising to resurrect it. So possibly in the next 5 years.
I think that's a really good assessment.
Their ultimate goal, to return it to its native environment.
It was the only marsupial apex predator. Bringing that animal back would have incredible benefits for the ecosystem.
But the $15 million project is already sparking fierce debate.
So many species in Australia are threatened with extinction. To spend a lot of money doing something that is unfeasible is a missed opportunity.
If they succeed, it will be a freak show. The animals will be so valuable. There's no way you can let them free range.
101 East travels to what was once thylacine heartland and talks to the scientists and their critics about the ambitious project to bring it back to life.
This specimen we've sampled in a few ways because we're working to try to optimize the best sources of DNA on the skin.
But it's this baby thylacine specimen that has proved the most valuable.
Here it is.
Yep, this is my favourite specimen of all of the ones that we have here.
More than 110 years old, it was preserved decades before the value of DNA was fully understood.
It was put into ethanol, which was quite amazingly fortuitous. What that did is it enabled it to preserve the DNA within that specimen really well. And so it's actually the one that enabled us to sequence the entire genome and will be the foundation of bringing the thylacine back.
But that's just the beginning. How to turn the genome into a living creature is another thing. In 2008, Pask's team had a major breakthrough. In a world first, they succeeded in bringing DNA from an extinct species back to life by inserting a thylacine fragment into a mouse.
We brought back a gene we thought was really important for skeletal development, for the shape and overall size of the thylacine. And we can tag the gene blue. So everywhere you see blue here is where you're seeing that piece of thylacine DNA, our Tassie tiger DNA, resurrected and actually functioning in that living animal.
Now they face a Herculean challenge, bringing back the whole genome of an extinct thylacine. And that's where this tiny marsupial, known as the fat-tailed dunnet, comes into play.
It's amazing to think a little marsupial like this could give birth to a Tasmanian tiger.
The dunnet is the thylacine's closest living relative. They intend to edit its genome to create a thylacine.
They're mostly the same. You know, we're talking 95% plus similarity between those two genomes, but there's 5% of difference. So what we do is we go in and we edit that 5%.
Stem cells will be sourced from the dunnet, then edited by Colossal to match the genome of the thylacine. The nucleus of a dunnet egg will be replaced with the nucleus of the engineered stem cell. The resulting embryo will then be implanted into its host.
One of the great things about marsupials is they all give birth to tiny, tiny babies. They're about the size of a grain of rice. What that means for us is that even that little mouse-sized fat-tailed dunnet can give birth to a baby Tasmanian tiger, even though it's going to massively outgrow the mum after birth.
These CT scans of rare baby thylacine specimens will then become critical.
We can actually map out their developmental trajectory to make sure that that final animal we get is going to be developing correctly along those pathways.
And if it's not,
then we would know that we're not recreating the thylacine, so we can stop those experiments, go back, have a look at what other bits we can change, and then create the next one and have a look at how that one's developing.
So really, until it's born, you won't know what you're going to get.
The whole goal with this project is to edit that genome to be 99.9% thylacine. But we don't know how big a difference that 0.01% might make, but it will definitely be a thylacine.
And definitely have stripes?
I hope so. I feel like we failed if it's not stripy.
According to Pask, it will be at least 10 years before a genetically engineered thylacine cell is produced. But Colossal CEO is much less circumspect.
We put a very big, ambitious goal out there for the mammoth of, 5 to 6 years. And elephants have a 22-month gestation. Given that our model organism, the fat-tailed dunart, has a 14-day gestation, which is obviously significantly shorter, I think that it's safe to assume that we will hopefully see one before we see a mammoth. That kind of gives you some idea of time.
So possibly in the next